So, once the muscle fiber is stimulated by acetylcholine, how do the structures within the sarcomeres generate the muscle contractions? This is the question that we're going to answer in this segment. To summarize, what we are about to briefly review is that the thick and the thin myofilaments within the sarcomere produce shortening of the sarcomere. And what this will do is it'll bring the Z discs that are at each end of the sarcomere closer to the center of the sarcomere. And formally, this process is called the cross-bridge cycle. So as we saw previously, an action potential that was created during excitation will spread across the sarcolemma of the muscle fiber. The action potential causes the release of calcium from the sarcoplasmic reticulum. That calcium will then bind to troponin, which, recall, was associated with the thin myofilament of the sarcomere. And this binding causes troponin and tropomyosin to shift position. And when troponin and tropomyosin shift position, what they will do is they will expose the myosin binding sites on actin. So now the myosin binding sites on actin are exposed, and the myosin heads of the thick myofilament may now bind to the thin filament. The binding of myosin to actin is what forms the cross bridge between the two myofilaments, and then the myosin will pull. This pulling causes the actin filaments to slide over the thick filaments, and in doing so, since the thin filaments are anchored to the Z discs, this causes the Z discs to be pulled closer to the center of the sarcomere. The sarcomere itself will shorten, which is going to generate the tension or the pulling force that we see in the sarcomere and in the muscle as a whole. Next, what we'll see is that ATP binds to the ATP binding sites located on the myosin heads. The myosin heads will then detach from actin, and what this does is that ATP energy functionally breaks the cross-bridge cycle. In rigor mortis, when someone expires, no new ATP energy is being created. So as we just described, ATP energy is not required to form the cross-bridge, but it is required to break the cross-bridge. So with rigor mortis, muscles will contract due to the formation of cross-bridges. However, the cross-bridges cannot be broken and muscles will remain stiff. The chemical energy of ATP is used to mechanically reset the myosin heads for another cycle. Figure 10.10 examines the sliding of the thin myofilaments over the thick myofilaments of the sarcomere in what is referred to as the sliding filament model of muscle contraction. The top diagram represents a sarcomere in a relaxed state. So notice that the small thin orange filaments, which are the tropomyosin proteins, are covering the myosin binding sites on the actin proteins, and the myosin heads that are in purple are unable to form the cross bridge with the actin filaments. In the bottom diagram, we see a sarcomere which is undergoing contraction, in which the myosin binding sites on the actin proteins are exposed, and the myosin heads are able to bind to the thin myofilaments and pull, which is going to bring the two Z discs at each end of the sarcomere closer together. So notice that neither the thick or the thin filaments shorten during this process. What does shorten is the distance between the two Z discs of the sarcomere. And this is ultimately what generates muscle tension, as there are thousands or even hundreds of thousands of sarcomeres within each muscle fiber. And when they're contracting, they would be doing this simultaneously.